Today, we will be discussing synapse physiology. We are going to start off by introducing the neuron. The neuron is unlike any other cell in the human body. It has a very distinctive shape which makes it very recognizable. Like most cells, it has a cell body. In the neuron, this is called a soma. Extending from the soma are branching extensions called dendrites, which receive information. The axon is a thin fiber that transmits electrical impulses away from the soma toward the next cell. The axon terminal made up of teledendria synapse with the next cell. There are two types of axons, the first being unmyelinated, which is pictured here. Myelinated axons have neuroglia that insulate the axon with a myelin sheet. The areas in which myelin are found on the axon are called internodes, and the unmyelinated spaces between are called the nodes of Ranvier. Now let's take a closer look at what's occurring at the end of the teledendria, at the synapse. The synapse is composed of three major parts: the presynaptic cell, the postsynaptic cell, and the synaptic cleft, which is the space between. The presynaptic cell is filled with vesicles. These vesicles are filled with neurotransmitters. For this example, we'll be using acetylcholine, which is going to be released into the synaptic cleft. Acetylcholine can either be excitatory or inhibitory. Generally, though, it is excitatory, meaning it will cause depolarization in the next cell. When an action potential travels down the axon to the synapse, it causes the opening of voltage-gated calcium channels, which allow an influx of calcium into the presynaptic cell. Calcium causes the vesicles to fuse with the plasma membrane, resulting in excitosis of the neurotransmitters. Let's take a closer look at what's happening at the postsynaptic cell. Located on the postsynaptic cell are chemically gated sodium channels, which have acetylcholine receptors. When acetylcholine is released into the synaptic cleft, it binds to the chemically gated sodium channels, causing them to open and allowing for an influx of sodium into the postsynaptic cell, depolarizing it. Acetylcholine is then removed from the synaptic cleft one of two ways: through simple diffusion or through the use of acetylcholinesterase, an enzyme which breaks down acetylcholine into acetic acid and choline. If a cell is depolarized, then an action potential propagates down its axon. Myelinated and unmyelinated axons use different forms of propagation. In an unmyelinated axon, there is a use of continuous propagation, which means that the action potential has to travel all the way down the axon. This makes this process very slow. Myelinated axons use saltatory propagation. That means that the action potential jumps from node of Ranvier to node of Ranvier, skipping the internodes. This makes this type of propagation very fast.